Okay, I'm gonna play the video. Let me know if you can hear it. Good.
A few years ago, Emmanuel Charpentier, I invented a new technology for editing genomes. It's called CRISPR-Cas9. The CRISPR technology allows scientists to make changes to the DNA in cells that could allow us to cure genetic disease. You might be interested to know that the CRISPR technology came about through a basic research project that was aimed at discovering how bacteria fight viral infections. Bacteria have to deal with viruses in their environment, and we can think about a viral infection like a ticking time bomb. A bacterium has only a few minutes to diffuse the bomb before it gets destroyed. So many bacteria have in their cells an adaptive immune system called CRISPR that allows them to detect viral DNA and destroy it. Part of the CRISPR system is a protein called Cas9 that's able to seek out and cut and eventually degrade viral DNA in a specific way. And it was through our research to understand the activity of this protein Cas9 that we realized that we could harness its function as a genetic engineering technology, a way for scientists to delete or insert specific bits of DNA into cells with incredible precision that would offer opportunities to do things that really haven't been possible in the past. The CRISPR technology has already been used to change the DNA in the cells of mice and monkeys, other organisms as well. Chinese scientists showed recently that they could even use the CRISPR technology to change genes in human embryos. And scientists in Philadelphia showed they could use CRISPR to remove the DNA of an integrated HIV virus from infected human cells. The opportunity to do this kind of genome editing also raises various ethical issues that we have to consider, because this technology can be employed not only in adult cells, but also in the embryos of organisms, including our own species. And so together with my colleagues, I've called for a global conversation about the technology that I co-invented so that we can consider all of the ethical and societal implications of a technology like this. What I want to do now is I want to tell you what the CRISPR technology is, what it can do, where we are today, and why I think we need to take a prudent path forward in the way that we employ this technology. When viruses infect a cell, they inject their DNA. And in a bacterium, the CRISPR system allows that DNA to be plucked out of the virus and inserted in little bits into the chromosome, the DNA of the bacterium. And these integrated bits of viral DNA get inserted at a site called CRISPR. CRISPR stands for clustered, regularly interspaced, short palindromic repeats. A big mouthful. You can see why we use the acronym CRISPR. It's a mechanism that allows cells to record over time the viruses that they have been exposed to. And importantly, those bits of DNA are passed on to the cell's progeny. So cells are protected from viruses, not only in one generation, but over many generations of cells. This allows the cells to keep a record of infection. And as my colleague Blake Wiedenheft likes to say, the CRISPR locus is effectively a genetic vaccination card in cells. Once those bits of DNA have been inserted into the bacterial chromosome, the cell then makes a little copy of a molecule called RNA, which is orange in this picture, that is an exact replicate of the viral DNA. RNA is a chemical cousin of DNA, and it allows interaction with DNA molecules that have a matching sequence. So those little bits of RNA from the CRISPR locus they bind to protein called Cas9, which is white in the picture, and form a complex that functions like a sentinel in the cell. It searches through all of the DNA in the cell to find sites that match the sequences in the bound RNAs. And when those sites are found, as you can see here, the blue molecule is DNA. This complex associates with that DNA and allows the Cas9 cleaver to cut up the viral DNA. Makes a very precise break. So we can think of the Cas9 RNA sentinel complex like a pair of scissors that can cut DNA. It makes a double-stranded break in the DNA helix. And importantly, this complex is programmable. So it can be programmed to recognize particular DNA sequences and make a break in the DNA at that site. As I'm going to tell you now, we recognized that that activity could be harnessed for genome engineering to allow cells to make a very precise change to the DNA at the site where this break was introduced. That's sort of analogous to the way we use a word processing program to fix a typo in a document. The reason we envisioned using the CRISPR system for genome engineering is because cells have the ability to detect broken DNA and repair it. So when a plant or animal cell detects a double-stranded break in its DNA, it can fix that break either by pasting together the ends of the broken DNA with a little tiny change in the sequence at that position, or it can repair the break by integrating a new piece of DNA at the site of the cut. So if we have a way to introduce double-stranded breaks into DNA at precise places, we can trigger cells to repair those breaks by either the disruption or incorporation of new genetic information. So if we were able to program the CRISPR technology to make a break in DNA at the position at or near a mutation causing cystic fibrosis, for example, we could trigger cells to repair that mutation. Genome engineering is actually not new. It's been in development since the 1970s. We've had technologies for sequencing DNA, for copying DNA, and even for manipulating DNA. And these technologies were very promising, but the problem was that they were either inefficient or they were difficult enough to use that most scientists had not adopted them for use in their own laboratories, or certainly for many clinical applications. So the opportunity to take a technology like CRISPR and utilize it has appeal because of its relative simplicity. We can think of older genome engineering technologies as, similar to having to rewire your computer each time you want to run a new piece of software. Whereas the CRISPR technology is like software for the genome. We can program it easily using these little bits of RNA. So once a double-stranded break is made in DNA, we can induce repair and thereby potentially achieve astounding things like being able to correct mutations that cause sickle cell anemia or cause Huntington's disease. I actually think that the first applications of the CRISPR technology are going to happen in the blood, where it's relatively easier to deliver this tool into cells compared to solid tissues. Right now, a lot of the work that's going on applies to animal models of human disease, such as mice. The technology is being used to make very precise changes that allow us to study the way that these changes in the cell's DNA affect either a tissue or, in this case, an entire organism. Now, in this example, the CRISPR technology was used to disrupt a gene by making a tiny change in the DNA in a gene that is responsible for the black coat color of these mice. Imagine that these white mice differ from their pigmented litter mates by just a tiny change at one gene in the entire genome. And they're otherwise completely normal. And when we sequence the DNA from these animals, we find that the change in the DNA has occurred at exactly the place where we induced it using the CRISPR technology. Additional experiments are going on in other animals that are useful for creating models for human disease, such as monkeys. And here we find that we can use these systems to test the application of this technology in particular tissues, for example, figuring out how to deliver the CRISPR tool into cells. We also want to understand better how to control the way that DNA is repaired after it's cut, and also to figure out how to control and limit any kind of off-target or unintended effects of using the technology. I think that we will see a clinical application of this technology, certainly in adults, within the next 10 years. I think that it's likely that we will see clinical trials and possibly even approved therapies within that time, which is a very exciting thing to think about. And because of the excitement around this technology, there's a lot of interest in startup companies that have been founded to commercialize the CRISPR technology, and lots of venture capitalists that have been investing in these companies. But we have to also consider that the CRISPR technology can be used for things like enhancement. Imagine that we could try to engineer humans that have enhanced properties, such as stronger bones or less susceptibility to cardiovascular disease, or even to have properties that we would consider maybe to be desirable, like a different eye color or to be taller, things like that. Designer humans, if you will. Right now, the genetic information to understand what types of genes would give rise to these traits are mostly not known. But it's important to know that the CRISPR technology gives us a tool to make such changes once that knowledge becomes available. This raises a number of ethical questions that we have to carefully consider. And this is why I and my colleagues have called for a global pause in any clinical application of the CRISPR technology in human embryos to give us time to really consider all of the various implications of doing so. And actually, there's an important precedent for such a pause from the 1970s, when scientists got together to call for a moratorium on the use of molecular cloning until the safety of that technology could be tested carefully and validated. So genome engineered humans are not with us yet, but this is no longer science fiction. Genome engineered animals and plants are happening right now. And this puts in front of all of us a huge responsibility to consider carefully both the unintended consequences as well as the intended impacts of a scientific breakthrough. Thank you.
Jennifer, this is a technology with huge consequences. You pointed out your attitude about asking, you can call it a pause or a moratorium or a quarantine, is incredibly responsible. There are, of course, the therapeutic results of this, but then there are the non-therapeutic ones, and they seem to be the ones gaining traction, particularly in the media. This is one of the latest issues of The Economist. Editing humanity is all about genetic enhancement, it's not about therapeutics, right? What kind of reactions did you get back in March from your colleagues in the science world when you asked or when you suggested that we should actually pause this for a moment and think about it?
My colleagues were actually, I think, delighted to have the opportunity to discuss this openly. It's interesting that as I talk to people, my scientific colleagues as well as others, there's a wide variety of viewpoints about this. So clearly it's a topic that needs careful consideration and discussion.
So there's a big meeting happening in December that
Correct.
You and your colleagues are calling together with the Academy of Science and others. What is your expectation? What do you hope that will come out of the meeting practically?
Well, I hope that we can air the views of many different individuals and stakeholders who want to think about how to use this technology responsibly. It may not be possible to come up with a consensus point of view, but I think we should at least understand what all the issues are as we go forward.
Now, colleagues of yours like George Church, for example, at Harvard, they say, yeah, ethical issues basically are just a question of safety. We test and test and test again in animals and in labs, and then once we feel it's safe enough, we move on to humans. And so that's kind of the other school of thought is we should actually use this opportunity and really go for it. Is there a possible split happening in the science community about this? I mean, are we going to see some people holding back because they have ethical concerns and some others just going for it because some countries under-regulate or don't regulate at all?
Well, I think with any new technology, especially something like this, there are going to be a variety of viewpoints. And I think that's perfectly understandable. I think that in the end, this technology will be used for human genome engineering. But I think that to do that without careful consideration and discussion of the risks and the potential complications would not be responsible.
No, there are a lot of technologies in other fields of science that are developing exponentially, pretty much like yours, and thinking about artificial intelligence, about autonomous robots, and so. No one seems, aside from autonomous warfare robots, nobody seems to have launched a similar discussion in those fields and calling for a moratorium. Do you think that your discussion may serve as a blueprint for other fields?
Well, I think it's hard for scientists to get out of the laboratory. Speaking for myself, it's a little bit uncomfortable to do that. But I do think that being involved in the genesis of this really puts me and my colleagues in a position of responsibility. And I would say that I certainly hope that other technologies will be considered in the same way, just as we would want to consider something that could have implications in other fields besides biology.
Jennifer, thanks for coming to that.
Okay. Okay, so I think also you can see that it's not only about like the biology of CRISPR, but the implications of using CRISPR, for instance, for like human enhancement and things like that. So I think the video is good, like to kind of get into what we will be talking about tomorrow, because you will be discussing this topic, as we said earlier. So yeah, but for today we can go back to science, and we'll do our experiment. I hope it works. It's the first time I'm doing it, so we'll see. Let me share my screen again. I'm going to share the presentation, and yeah, and we can go over that. But so far, do you guys have any questions, any comments, something that wasn't clear? ¿Alguna pregunta, algo que quieren que les aclaremos? ¿Está todo como, más o menos están entendiendo el concepto de CRISPR? ¿Algún comentario o algo antes de comenzar? ¿No? Bueno. Todo bien, muy bien. Not yet, dice Gabriel. Sí, ahora capaz van a tener un poco más de preguntas cuando empecemos con el laboratorio. Okay, let's do this. 1 second. Okay. Okay, there you go. This one, we can delete the slide. Okay. Good. I will put you guys here. So everyone sees my slides and can hear me well and everything good?
Yes.
Okay, perfect. So I will do it in Spanish and in English, okay? So we wanted to, like, the term to be clear. Sometimes like it's better to repeat and repeat until it's really clear so you can like understand. So kind of like to go over what we discussed last class, we can see here how the CRISPR system works and how it comes from bacteria as a way to defend themselves from the attack of certain viruses in these that are called bacteriophages. So we will like repeat it again. So when the virus attacks the bacteria, the bacteria has a CRISPR RNA that can bind specifically to the genetic material of the virus, and then when it binds the Cas9, which acts like a molecular scissor, it's going to degrade the DNA of the virus, or the RNA of the virus, and then it will lead to destroying the virus, and then the virus cannot replicate. Entonces, cuando hemos hablado de los bacteriófagos, cuando infectan a las bacterias se van a defender utilizando estos CRISPR RNAs que se van a pegar al material genético del virus, y entonces la Cas9, que es como una tijera molecular, va a cortar el DNA del bacteriófago, y entonces el bacteriófago se destruye y la bacteria gana. Otro concepto que creo que va a ser importante para que entiendan el laboratorio de hoy es lo que hemos hablado acerca de el lag operón, que lo hablamos en la primera clase, no sé si se acuerdan. Si alguien me puede un poco explicar lo que habíamos hablado acerca del lag operón. So we talked about the lag operon last class. I hope you remember a little bit. So maybe someone wants to explain what they remember about this system to the class. We talked about sensors and how the bacteria prefers to have glucose other than having like lactose as like a, right? Who wants to explain? No one? So we talked about gene regulation. Mari Carmen, I think you were about to do it.
Yes.
Oh.
Well, the lack of operon was that when there's glucose in the ambient, I guess, it is activated, so the bacteria can metabolize glucose. And when there's no glucose, it's deactivated. It was something like that.
Okay, yeah. So when it has to use another source of energy, then it will activate these genes here, right? That will allow the bacteria to metabolize another type of sugar. And so these genes will help the bacteria do this. So remember how some like sugars are composed of like two different like two types of like mono sugars, and then they can be broken down into two. So this gene, the LAC-C, is going to help the bacteria hydrolyze this other energy source. Okay, is that clear? En español, habíamos hablado del lag operón en la anterior clase y Mari Carmen estaba explicando que la bacteria prefiere usar azúcar, pero cuando no está el azúcar y tiene que usar otra fuente de energía, entonces puede activar este lag operón, y algunos genes, por ejemplo el LAC-C, va a ayudar a romper el azúcar en dos para que la bacteria lo pueda metabolizar. Entonces, this system had two main components that we talked about. We mentioned about how a gene can be induced, right, when there is an activator. And then also we talked about how a gene can be repressed when there is like a repressor, right? So only when the repressor goes away and the activator is present, then the gene can be turned on. Okay, is that clear? So in Spanish. So cuando el gen se activa, solamente cuando el activador está presente y cuando el represor está fuera del gen, porque si no va a bloquear la transcripción de este gen. ¿Ok? Entonces eso tiene que estar bien claro porque es parecido a lo que vamos a hacer. ¿Ok? Okay. So for this experiment, what we will do is we will introduce a mutation in the beta-galactosidase gene, or beta-gal gene. Okay, what does this gene do? When this gene is activated, and then you can like the bacteria can break down this analog, which is a lactose analog. And then when it breaks it down, this colorless metabolite will become blue. Okay? Is that clear? Entonces, en español, la bacteria tiene cuando está la ex-gal, que es un análogo incoloro de la lactosa, como tiene el gen del LAC-C, it has this gene that is intact. This gene will produce a protein, we have talked about this, and then this protein, esta proteína va a cortar este análogo de la lactosa, y entonces se va a volver, era antes incoloro y ahora se va a volver de color azul. Entonces, las bacterias que tienen el gen del LAC-C y que tienen ex-gal son azules. ¿Sí? Si nosotros introducimos una mutación en el gen del LAC-C, entonces, ¿qué quiere decir? Que no se va a producir la proteína beta-galactosidasa y por lo tanto, entonces, a pesar de que haya el metabolito, esta bacteria no va a tener color, va a ser blanca. ¿Por qué? Porque no tiene la enzima que produce el cambio de color. Okay, is it clear? So in the case of these bacteria, when we will introduce a mutation with CRISPR, okay, so it will render a non-functional protein. So this means that even though the ex-gal is present, but the gene is not active, right? So this bacteria will be white, and then this bacteria will be blue. Is that clear? Did everyone understand?
I had some trouble with my internet, but what I understand is that they will be introduced a mutation in the beta-gal gene.
Yes.
And this one breaks the ex-gal that it's like lactose.
Yes.
But why it's blue?
Okay, so because when you break down like the ex-gal, one of the subproducts, it's blue. It's like it becomes blue. It's a way to phenotypically see that we have introduced a change, right? Sometimes when
okay,
like a chemical compound and then it changes its structure, it can also like change color. So this will be our way to know if the LAC-C gene is working or not. For the sake of the experiment, we want to know if we were able to introduce a mutation. So the cells that have introduced the mutation will be white, and then the other ones will be blue. This ex-gal, I can show you. Yeah, well, maybe this is better. So this beta-galactosidase, the gene is going to catalyze the conversion of this substrate, and then when it changes the structure, then it becomes blue. Okay, so the cells will be blue. But when we don't have a functional beta-gal, then this reaction doesn't happen, and then these cells are white. Is it clear?
yes.
Just to be clear, the LAC-Z is the actual gene sequence, and when the promoter becomes activated, that's gonna transcribe or translate into the protein, which is the beta galactoside. So just remember, LAC-Z is the gene, the beta gal is the actual protein that breaks down lactose. But like Yara says, we can't really use lactose because we're not going to be able to physically see if it's breaking down. So what they do is they use like an analog, which in this case is the ex-gal, which gives you a color. So when you have a functional gene like that's complete sequence, this means that the E. coli is going to use this machinery. Like we'd learn about the central dogma, it's going to transcribe the gene, then translate it into this beta galactoside protein, and that's going to break down ex-gal, the analog, which is going to give us blue. And if the gene is non-functional, then you're not going to be able to translate into this protein, the beta galactosides. And if you don't have this protein, then you're not going to be able to break anything down, which means that you're not going to be able to break down the ex-gal that gives you blue. So you're going to have a white color, if you want to think about it that way.
Yes, that's right. Sometimes the gene can be like confusing, the gene name, because it's not exactly the same. But this gene gives rise to this protein, right? here like the gene is working perfectly, but when we CRISPR it, we will introduce a mutation here, and then the mutation will make this gene non-functional. So we won't have the protein. And if we don't have the protein, no one is able to do this reaction. And then these cells are white. ¿Sí? ¿Se entiende? ¿Están todos claros?
Yes.
¿Puedes volver al primer slide, Yara?
Sí.
El otro. No, el primero que tenías. No, ese de ahí. O sea, si entienden aquí, like if you guys see here, you guys know what a promoter is, right? A promoter is what initiates translation of a gene, right? We're gonna go from RNA to protein. Entonces, when you have like a lactose, or in this case, the analog, the X-gal, into your culture, the lactose, I believe it binds into this, correct me if I'm wrong, Yara, but it binds to this repressor. So usually this repressor.
Yeah, no, but that is not the X-gal. So the X-gal is the substrate of the enzyme.
Sorry, yeah.
IPTG, yeah, to
yeah, so we want to activate that what I showed at the beginning. We want to activate that in order to get
so we
want to activate all the system in order to get this gene active.
Yes, that's right. So that's another component so that we have in this equation. So this gene is not going to be on by itself. It needs to be activated, and to activate it we have to add a substance that is called IPTG o IPTG en español. Entonces, para activar el gen tenemos que tener este activador. Entonces tenemos que agregar IPTG. IPTG se une a esta proteína que es la activadora, y entonces enciende la transcripción de este gen. Entonces necesitamos dos cosas so far. We need two things, right? We need to have the active gene, so we get the protein, and then we get the phenotype change of color. And we also need to have the IPTG, because if we don't have IPTG, what happens? This gene is not going to be transcribed. Even if it is the right sequence of the gene, even if the gene is not mutated, we won't have it on because we need the activator. Okay? Is that clear? So before this step, we even have like here a promoter, and we need to activate the gene. When we activate the gene, then we get the beta-galactosidase, and then we get the blue. But if we don't have the IPTG, this gene the cells will be white, right? Because the gene won't be on, even if it is not mutated and if it's a right gene. Is that clear? ¿Está claro? ¿Alguien que quiera explicar para los compañeros, para ver si estamos todos en la misma página? Porque se va a complicar un poquito más, entonces I want to make sure that it's clear.
Aquí algunos en el chat dicen que sí entendieron. A ver, Pedro dijo que sí entiende. Do you want to explain? Fabricio, Joana, los que dijeron que sí entienden, ¿quieren explicarlo otra vez para los compañeros que no?
I could try to say it in English.
Go ahead, yeah.
So correct me if I'm wrong, but with the bacteria with the LAXI gene active, we're going to need to add a protein, like it's the promoter to activate it. So it is going to generate a protein that it is the big O, and this protein is going to break the X-gal, which is the analogous of the lactose, right?
That's perfect. So what is the goal of this experiment? What are we trying to do?
We're going to add the mutation to inactivate the LAXI gene. So when even in the presence of the X-gal, it is not going to translate the protein, transcribe the protein, so there's not going to be any reaction. So it's going to stay in color.
Muy bien, perfect. So we will start with blue cells, and if we are successful and we introduce the mutation with CRISPR, then the cells will turn white. ¿Sí? Entonces vamos a usar CRISPR para generar esta mutación. ¿Sí? Y si lo logramos, entonces estas bacterias ya no van a ser azules, van a ser blancas. Vamos a empezar con bacterias azules y vamos a terminar con bacterias blancas.
Alguien en el chat pregunta, Ángel pregunta que entiende que el IPTG activa el sistema, pero ¿qué es exactamente el IPTG? Pregunta.
El IPTG es una sustancia química, es como una sustancia así como estas que ves aquí. No es una proteína ni nada de eso, es simplemente como una sustancia química que activa, que se une al activador y entonces juntos se van al promotor. ¿Ok?
Y Jessica pregunta...
And add it to the plate.
Y Jessica dice, ¿se desnaturaliza o por qué termina blanca? Fabricio dijo que lo puede explicar en español. A ver, Fabricio, si puedes explicar en español para los que hablan español.
Bueno, lo que entendí es que ahí lo que hacemos es lograr una mutación y bueno, ahorita me acabo de confundir porque creo que escuché que cuando vamos a hacer la mutación va a salir en blanco. No sé si estoy bien o mal.
¿Por qué va a salir blanca? Ajá.
Porque no va a poder producir lo que es el vegal, la proteína.
Ajá, exacto.
Y en cambio el otro sí lo produce, por lo tanto van a salir en color azul.
Exactamente. What's wrong here? Ok. Sí, exactamente. Entonces no es que se va a desnaturalizar la proteína, simplemente la reacción no se va a dar, ¿verdad? Porque había ex-gal y entonces si está la proteína, we go back to this one, si está la beta-galactosidasa, oh, what's wrong? I wish I knew my password.
Oh, no.
Okay.
O sea, si piensan, a mí siempre me gusta repetir acerca del dogma central. Si piensan en un gen, if you have a sequence of DNA, para cualquier proteína en su cuerpo, o en el E. coli, ya hablamos de eso el día 1, cuando tienes una secuencia de ADN, para la insulina, por ejemplo, y si necesitas insulina, ¿qué pasa? You have your DNA, and that's going to be what? Transcribed into mRNA, right? Va a ser transcrita al RNA, y luego de ahí va a ser traducida a una proteína, y esa proteína va a hacer su función, sea lo que sea. En este caso estamos haciendo eso, tenemos el gen LAC-C, que está perfecto, secuencia perfecta, tiene una función de producir la proteína GAL. ¿Para qué? Para que metabolice la lactosa o aquí el análogo. Y ahí vamos a obtener un color azul en las colonias. Ahora, si le metemos CRISPR a esta secuencia de LAC-C, si lo interrumpimos de alguna manera, hacemos un corte y creamos una mutación, ¿qué va a pasar con esa secuencia? Esa secuencia ya no va a ser perfecta. Entonces esa secuencia no va a ser, o sea, no va a avanzar, no va a ser transcrita al RNA ni traducida a proteínas. Entonces va a quedar trancada ahí, va a ser non-functional. Entonces, if it's non-functional, no va a poder producir la proteína de galactosides. Y si no tienes esa proteína, no puedes, ¿qué? No puedes metabolizar la lactosa. Entonces, por eso que vamos a tener las colonias blancas.
Exacto. Okay, is it clear? We'll go to the next step and things will get a bit more complicated. But if you are understanding step by step, then you should have no problem. Okay, so The goal of the experiment, we will repeat one more time, then it's to introduce a mutation in the LAC-Z gene so that the X-gal that doesn't have any color like cannot be metabolized. And if it's not metabolized, then the bacteria will be white. Because if the gene is functional, the bacteria are blue. Okay, this I think it's clear. Okay. So this is the bacteria that we'll use for the experiment. It's called E. coli HB101 PBR-CAN. I think CAN doesn't matter. It's because of kanamycin resistance. So if we want to do CRISPR, right, we talked about like what we need in the other class. So we of course will need the in this case it's not the Cas9, sorry. I use this Cas9 a lot in my experiments, so I put it like that. It's actually just Cas9. Okay, so we will need the Cas9, right? We need the LAC-Z, and then we will need that the bacteria is able to do recombination, right? Or repair. Otherwise, if the Cas9 cuts the DNA, the bacteria dies, right? So can someone explain what we will use each component for? So what is the Cas9 for? Why do we have the LAC-Z, and why do we need the homology-directed repair in the bacteria? I kind of already gave the answers, but maybe someone can clarify. ¿Alguien que pueda explicar por qué necesitamos estos componentes para hacer CRISPR?
LAC-Z is the gene sequence that we are going to add the mutation in. Cas9 is the one that it's going to target the sequence of target the sequence of the gene.
It's the one that will cut.
The one that will cut. And the plasmid is where the gene is going to be, right? Or no?
And the plasmid, and yeah, well, we could have LAC-Z in a plasmid, that's true. But in this case, we are talking about a plasmid to do homology-directed repair, right? Maybe we can I can explain a little bit better.
No entiendo por qué necesitamos un plásmido para hacer recombinación homóloga si queremos hacer una pérdida de la función de la enzima, entonces se podría hacer con una deleción.
Exactly, but what happens, we talked about this, I think, we talked about this, right? If we cut the bacterial DNA, the bacteria dies, right?
Nos habías dicho que la célula improvisaba, ¿no? Y así como tenían la fractura, pues seguía con el ciclo.
Yes, that's true. Puede improvisar, pero tiene que tener algún sistema de reparación para improvisar. Si no tiene ningún sistema de reparación, no puede improvisar y se muere.
¿El plásmido contiene las enzimas para reparar el corte?
Exacto. El plásmido exacto, ¿sí? Y en este caso, esta bacteria que hemos comprado está como que genetically engineered para poder hacer solamente esta, que es la homology directed repair, ¿sí? ¿Por qué creen ustedes que esa bacteria a propósito, we don't want it to do the non-homologous end joining? Why do you think we don't want a bacteria that can do this type of error-prone repair?
Para que se pueda hacer la mutación y ella no vuelva a ser como era antes.
Exacto, because if the bacteria has non-homologous end joining, what is it going to do? It will generate random mutations, right? And it can be dangerous, right? Like what if the bacteria makes a mutation that for some reason allows the bacteria to be resistant to an antibiotic, which is unlikely, but it could happen. So we are using this technology to learn something, right? And if it cannot be dangerous, we cannot generate like a bacteria that it's going to do its own repair, because we don't know which type of mutation it will generate. And it could be a mutation that can alter how bacteria responds to antibiotic. And without us wanting it to happen, we could create some super bacteria or something like that. Is that clear? Si la bacteria tuviera la manera de hacer non-homólogos en joining, cuando nosotros cortamos con CRISPR, la bacteria va a generar mutaciones aleatorias, porque hemos hablado de que va a intentar juntar esas dos partes del ADN y sobrevivir. Pero esto puede generar una mutación que le dé alguna ventaja a la bacteria, digamos de resistencia a antibiótico o cualquier cosa, y nosotros estaríamos generando alguna bacteria que podría ser peligrosa si la tenemos en el ambiente. Es poco probable que pase, pero para hacerlo de manera más segura es como un safety log, ¿no? Es como que a prueba de niños. Entonces, la bacteria no puede hacer esta recombinación, pero sí puede hacer esta, porque nosotros le vamos a poner un plásmido que específicamente puede hacer homology directed repair. Entonces nosotros le vamos a dar a la bacteria, ¿sí?, un donor DNA para que haga la mutación o la inserción que nosotros queremos, no lo que la bacteria quiere. Because if the bacteria does it by itself, it could generate an unwanted mutation, right? You see, it can be like kind of dangerous. But if we tell the bacteria, we give it a template, we will make the bacteria do exactly the mutation that we want. It cannot do this, okay? This bacteria is like a special bacteria that cannot do that. It can only do this. Is that clear?
Jess dice que she's lost. ¿En qué no entiendes, Jessica?
I wasn't like following why we were using the homology directed repair, but I think that I now understand.
So this cells were, I guess, in the company, their mechanism was hijacked so that the non-homologous end joining would like not be expressed, like that machinery wouldn't be expressed at all. So that's completely shut down, even though they could, you know, but they're, I guess, they have been in a way modified where they cannot express that. And remember I talked about plasmids on the first or second day of class last Sunday. We said that plasmids are a circular DNA or genetic material that some bacteria have aside from their bacterial DNA. And we nowadays, you take advantage of these plasmids or vectors, and we use them as vehicles or like as transporters to like add desired DNA sequences. Remember we talked about the restriction enzymes and all that, and the circular plasmid where we add desired sequence. So in this case, that's what they introduced into this commercial bacteria in the whatever company it was. They designed a plasmid and they introduced all of this sequence for I guess the proteins, the sequences needed to do this homology repair mechanism. So that's what it pretty much is. So you have inside the E. coli, you're gonna have that plasmid that I showed last time with the desired sequences to take care of the HDR, if that makes sense.
So like in theory, right? In theory, we could use non-homologous end joining if our goal is to disrupt the gene, right? We just want it to be non-functional. So this system, you could think it's the best one to achieve that goal. The problem is that if you leave the bacteria to generate like any mutation that the bacteria wants, by chance, very low chance, but it could happen, the bacteria can generate something that we don't want or we don't know what it's doing, and it can be dangerous if we release that bacteria into the environment, right? It's just like to be safe. So we will direct that bacteria, and we will give the bacteria a donor DNA so that the mutation that we, or the insertion that we will generate, it's going to be decided by us. It's not the bacteria that will do whatever it wants to do, but it's us. We will let the bacteria know exactly which mutation we want. It's just to be safe. Okay, is that clear?
Yeah, thank you.
Yes, I have a question. So non-homologous end joining is not very used because of the risk of that that it can choose like whatever sequence. And here it says like there's the deletion and then the insertion. So the insertion is by luck, by chance.
So, okay. So yeah, in this case, in this specific case, we are not going to use this, right? But we could, like we've seen when we talked about, when we did the groups last week, some of you had a disease that needed the gene to be inactivated. And the right way to do it was this, because you just want to inactivate the gene, right? In this case, for our lab purposes and for safety reasons, we are doing it in this way, okay? So we will provide the bacteria with a donor DNA. We will give the bacteria, and then this will generate an insertion, right? It's not gonna be random because we will use this system that is not random. So we know the sequence of the donor DNA. We will provide this to the bacteria, and the bacteria will insert this in the gene. So what happens is that what will happen is this, right? We will have an insertion, and then the gene won't have the sequence that it used to have, and it's not going to be able to be transcribed and then translated into a protein. But we will know what happened here. We will put this donor plasmid as part of the experiment. Okay? It's true, if we wanted to just disrupt the gene, we might as well use the other way. Like, we don't need to provide a sequence because we just want to disrupt it. We could just cut and let the bacteria deal with the cut. And for sure, it will generate a mutation that will render the protein non-functional. But in this case, we are just, for safety reasons, we will use like the other way. That we could like, we could even introduce some other gene that we want in this way, right? We could also do that. But in this case, we are going to use this to disrupt the gene of the beta-gal, and then we will see the effect on the phenotype, because the bacteria will change the color. Is that clear?
Yes.
Okay. So what happens if think about this, like what happens if the bacteria doesn't have any way of repairing the DNA, and we just introduce the Cas9, and but it doesn't have the plasmid for homology directed repair, and it also cannot do non-homologous end joining. What will happen to that bacteria?
It is dying, right?
Exactly, it will die. Why? It will die because like it will generate cuts, right? And then the bacteria cannot, no one can live with a cutted DNA. It's not compatible with life, okay? And we will see that as well. Part of the experiment, we'll see. Okay, so far, is it clear? Are we good?
If it's not clear to anyone, just, it's okay, you guys can say so.
Yeah, it's like it's kind of like complicated. So it's, don't feel bad if you have doubts. Like, it's a complicated process. Okay?
Everyone's got cool.
Okay, it gets a little bit more. We'll go one step further, okay, now. So up to now, what we know is that we will start with blue bacteria, right? Empezamos con bacterias azules. We will disrupt the LAC-Z gene. Vamos a generar una mutación en el gen de la beta-galactosidasa. And the bacteria will turn from blue to white, right? We talked about the activator that will be IPTG that will bind to the promoter of the beta-gal, right? We talked about this promoter here, IPTG, to drive the expression of this gene. But remember what Soa told you last class. We are introducing another plasmid with homology-directed repair. And guess what? This plasmid also needs to be activated, right? It needs to be activated. And how do you activate that plasmid? You activate it with arabinose, okay, which is a sugar. And then only when you activate the homology-directed repair, the plasmid will turn on this. If it's off, if you don't put arabinose, then the homology-directed repair, or HDR, won't be turned on. Therefore, the bacteria will die. Even if it has the plasmid, if it's turned off, it's going to die because it doesn't have the activator, which is arabinose. Is that clear? So we have two plasmids, right? We have one that could be for the beta-gal that will be activated with IPTG. And then we have another plasmid with the DNA repair system that is activated with the arabinose. We need the repair system so that the bacteria can fix the problem when we cut with the Cas9. Okay, are we clear so far?
Yes.
See? Okay.
Can you repeat it again, please?
Sí, claro. We have two different plasmids, right? We talked about the LAC-Z that has its own promoter. Promoter means like the activator region to turn on the gene, okay? That's going to be activated with IPTG. But on the other hand, we have another plasmid that has the DNA repair system for the bacteria. And this plasmid also has its activator, right? And the activator in this case is not IPTG, but it's arabinose, which is a type of sugar. You can just add that to the culture, and then the bacteria will turn on this gene. You need it to turn on this gene. When there is no arabinose, then this plasmid is not being active, and then the bacteria cannot activate the DNA repair system. Is it clear?
Yeah, thank you.
Sí, para todos los demás es complicado. Yo sé que es complicado, pero así pasito a pasito yo creo que podemos entender.
One question. E. coli have has the plasmid with LAC-Z because it doesn't have the LAC-Z gene integrated in its genome, like.
Yeah, that's a good question. It could be that the like we talked about, like the E. coli, that it's a natural thing for the E. coli to use this system whenever they want to, they don't have glucose, right? But in this case, we are using a commercial bacteria that is just tailored for us. So like they put like this plasmid is a genetically modified bacteria, so that they put like this plasmid that is induced by IPTG, and all of these things are like made so that we can use the bacteria. It's not like a bacteria that you will find in like the environment, right? It's just for like learning purposes that we use this commercial bacteria. like CRISPR, right? CRISPR was discovered in bacteria, but we are also adding the Cas9 to the system. It's just because we want to be able to put each component and then manipulate everything so that we know what will happen for our experiment. Okay? Okay, so this is the last part of that. If you understand this last part, then you will be able to make hypotheses of what you think will happen in different conditions, okay? So we talked about the homologous recombination, right? And we said we will have this system, right? We will use this system today. So we need to have a donor plasmid, right? We need to have a donor DNA. Otherwise, the Cas9 won't be able to do its job because it doesn't have the donor DNA, okay? So we will put that in form of a plasmid. We have two plasmids, the control plasmid, okay? And then we have the donor guide. This one will be like just a control. So it will be used by the HDR to double to impair the LAGC gene function, right? And then we'll have this other one, which includes both the donor template DNA sequence, okay, and the sgRNA, because the sgRNA will guide the Cas9 where it has to go and do the insertion. Okay, is that clear? This will be just a control plasmid with no guide RNA. So this one won't be completely active because it doesn't have the guide RNA, right? But the donor guide plasmid, it has the guide RNA and it has the sequence so that it will go where it needs to go. The guide will guide the Cas9 to the place that we want to make the mutation. So this will be a negative control because this won't have the guide RNA. So the bacteria will not know where to make the cut. Is that clear? It's just a control. Entonces, en este caso tenemos el control que solo va a tener el donor DNA, ¿sí? Pero no tiene el sgRNA. Entonces, sin el sgRNA, la Cas9 no funciona. Acuérdense que CRISPR-Cas9 necesitas tener Cas9 y necesitas tener el guide. Si no tienes al guide, la Cas9 no sabe a dónde ir y entonces no va a cortar, ¿sí? Por más de que tenga el donor template, no va a cortar porque no tiene el guide. Y este otro plásmido tiene el guide RNA y tiene el donor DNA. Entonces, este es el que funciona, este no debería funcionar. Si ponemos este plásmido, entonces las bacterias no deberían cambiar de color porque no tienen el guide RNA y la Cas9 no funciona. En este caso, todo el sistema está completo y entonces sí debería cortar. ¿Sí? Is that clear?
Entonces, básicamente, estos son otros sets de plásmidos que estamos metiendo, igual que cuando metimos el plásmido que tenía las secuencias necesarias para llevar a cabo el homology direct repair. Ahora queremos introducir, ¿se acuerdan cuando hablamos de CRISPR la pasada semana que dijimos que necesitamos para hacer un experimento de CRISPR? ¿Se acuerdan? ¿Para qué necesitamos el sgRNA?
To guide the Cas9.
To guide the Cas9, exacto. Entonces necesitamos una secuencia de ARN para guiar al Cas9 y decirle, Cas9, tienes que cortar acá. Puf, él lo corta. Pero en el caso del primero, si solamente es el donor DNA, el insertion que queremos meter, pero si solamente es eso, ¿dónde va a ir con el Cas9? A ninguna parte, porque el sRNA no está ahí, entonces no hay nada que guíe al Cas9 para que corte. Entonces, por eso, como Yara dice, ese es el control, donde capaz no pase nada porque no hay corte. Y en el segundo plásmido, ahí sí tenemos, o sea, piensen como lo explicamos la semana pasada, ahí sí hay un sRNA, ahí sí ya tenemos el Cas9, obviamente, en el E. coli, y tenemos un donor template de DNA, que son las 3 cosas esenciales que necesitamos para hacer un CRISPR. ¿Entendiste, Gabriel? ¿Is that clear, Gabriel? Gabriel estaba preguntando si lo podemos hacer en español. Más o menos, que no entiendes.
A ver, capaz ahora va a quedar un poco más claro. Let's
Sí, sí, sí.
Let's do the next one. Okay.
Right.
Now we put it
Tengo
together.
una pregunta.
Sí.
¿A partir de dónde se transcribe el Cas9?
La bacteria que vamos a usar
Bueno,
ya tiene Cas9. We won't put it.
gracias.
Podríamos ponerla como otro plásmido, pero estas que hemos comprado ya tienen Cas9. Ya tienen Cas9 y ya tienen el plásmido de la Homologous Recombination Repair. Nosotros lo único que vamos a hacer hoy es poner los plásmidos estos del donor, el control y el donor plus guide. Porque las bacterias que tenemos, ¿sí? Ya tienen el repair system, pero tiene que ser induced by arabinose, right? Is that clear? So I know it can get a bit confusing, but let's go one by one. And let's try to think what's going to happen in each case. Okay, so we have here the blue bacteria that we will start with, right? The bacteria, all the bacteria have already the Cas9, right? In this culture media, we have no arabinose. So that means that the repair system is going to be off. Okay, is that clear? So if we put here only the donor plasmid, what do you think is going to happen? Which color of bacteria are we going to get?
White ones.
White? Why? Why do you think it will be white?
I think that if they were white, they will keep white.
No,
I
they are blue.
don't know if I had get confused about the color of the bacteria. It was blue or white in the beginning.
We start with blue bacteria.
Okay. Then it will continue blue.
Okay, it will continue to be white. Why?
No, you said that the bacteria was blue. Then
Why do you think it will continue to be blue?
Because there is no air, then the plasmid won't be.
The plasmid what?
You're cutting off, Fernanda.
Won't be transcribed. No será transcrito. No sé.
Okay. Okay, so Fernanda says that the plasmid, which plasmid? We have two plasmids, remember. We have one for DNA repair, and then we have one with the, well, actually 3 plasmids. We have one with the with the DNA repair system. We have one with the donor, and then we have another plasmid with the donor plus the guide. So what's going to happen in situation A, B, C, D? Which color of bacteria, or what are we expecting to see? So Fernanda says this will be blue, right? It will be blue because, can you repeat why do you think it will be blue?
Yeah, it's going to be blue.
O sea, yo también estoy tratando de entender, a ver. El primero A es el E. coli, que solamente tiene el Cas9 y el plásmido que es con el donor, ¿no?
Ajá.
Ok, entonces esta bacteria solamente tiene Cas9 y el plásmido con el insert. Entonces aquí Fernanda dijo que aquí van a continuar siendo azules. ¿Por qué? Tienes Cas9 y solamente el donor DNA, nada más.
Because we don't have the guide, so Cas9 will not recognize the sequence, and it's not going to happen the cut and the repair.
Okay, that's a good point. So let's discuss then B. In B, we have Cas9 and we have the donor and the guide. Are these bacteria going to be blue, white? What's going to happen here?
I think that the bacteria is going to die
Why?
because it has no arabinosa, so it's not going to repair it after the cut. And you have the donor guide with the sgRNA, which is going to guide the Cas9 to cut the lexin.
Did everyone understand what Pedro said? Pedro dijo que en el caso B, a pesar de que tienes el donor y el guide y la Cas9, las bacterias van a morir. ¿Por qué? Porque no tienen el repair system. ¿Y qué hemos dicho que pasa cuando no tienen el repair system? Corta, va a cortar porque tiene el guide y tiene todo, tiene Cas9, tiene el guide, va a cortar, pero se van a morir porque no van a poder reparar el daño. ¿Sí? Entonces, en este caso, como no hay arabinosa, recuerden que la arabinosa activa el repair system. ¿Sí? Entonces, como no hay arabinosa, estas bacterias se van a morir y entonces no deberíamos ver nada. Is that clear for everyone? So here we will see blue bacteria. We expect to see blue bacteria because we don't have the guide. So the Cas9 doesn't know where to cut, right? It won't cut. In this case, we are expecting to see no bacteria at all because we don't have the repair system. Even though we have everything else, we don't have the repair system, so the bacteria is going to die. ¿Sí? ¿Está claro?
Bien, chicos.
Entonces, ¿qué va a pasar en C?
I think that it will continue to be blue. Even with the repair system, there's not going to be the cut because we don't have the sgRNA. Since the plasmid is the donor, the control.
Perfect. Okay, is that clear? En C van a seguir siendo azules. Es casi lo mismo que en A, ¿verdad? Pero la diferencia entre esta y esta, aparte del repair system, es que aquí no está el guide RNA. Entonces la Cas9 no va a cortar, porque no está su compañero, su amigo, que es el guide. Son súper amigos, van juntos a todo lado y como no está, no quiere cortar. ¿Ok? So someone else tells me what will happen in D. ¿Alguien más?
In D, it will be blank because it will cut and then repair.
Yeah, so we are expecting to see white bacteria, right? If everything works and if CRISPR works and we are successful, then we should see white bacteria because we have all the things we need to do CRISPR. We have the repair system, we have the guide, we have the donor sequence, and we have the Cas9. So here we should see white bacteria. Okay?
I have a question.
Yes.
In these experiments, we still have the possibility to deal with off-target cuts or no?
Off-target effects of the
Cas9.
Of the Cas9. Yeah. It could be. I mean, we don't know. It could be that we have off-target effects. To know, we would have to sequence the bacteria and then see how the sequence has changed. But we are using homologous recombination repair to kind of like guide and make the mutation that we want. So, and to decrease the possibility and the likelihood to make an off-target effect. But I cannot say it's not going to happen, right? We don't know. We would have to sequence to know for sure.
Yeah,
But
good.
very unlikely. Okay, is it clear to everyone? Sí. Okay.
Yes, clear.
Bueno, entonces we can now go ahead and then we will do the experiment. I will give you the link to the guide, to the student guide, so that you know what we will be today. I will let you know what I have done so far. Okay. So far, what I have done is there is one little another thing more to consider. Okay. So how do we know that today we will transform this bacteria with these donors and then with these plasmids, right? This is what we are doing today. But how do we know that we were successful and that the plasmid got inside of the cell? Maybe something went wrong and it doesn't go inside of the cell, and then the plasmid is not there. How can we make sure the plasmid is inside of the cell?
Some slides ago, there was information that the plasmids were resistant to a medicine. I don't know which one is it. So after we insert the plasmid, we're going to kill the ones without it. Is it right?
Okay.
Select the ones.
So I talked about this in the class before. She talked about selection. So vamos a seleccionar. Estos plásmidos van a tener un gen, además de resistencia a un antibiótico. Entonces, solo las bacterias que han podido meter el plásmido adentro van a poder sobrevivir en ese medio de cultivo con antibiótico. Las que no tienen el antibiótico se van a morir porque no pueden, no sobreviven, no son resistentes a ese antibiótico. Entonces estos plásmidos todos tienen resistencia a un antibiótico que se llama streptomicina. ¿Sí? Entonces las placas en las que vamos a usar, ¿sí? Tienen todas streptomicina. I don't know if I have, yeah, here. Okay, the antibiotic resistance. Otherwise, it could be that the transformation is not successful, right? We won't know. We wish we could have super eyes and then see the bacteria and see if the plasmid is inside, but we don't. So we need a way to see it, to know the phenotype. So when we select with the antibiotic, we are sure that the transformation was successful. Is everything clear? Like, is everything together? Today we will be doing this part. We will be transforming this bacteria, okay? We'll transform the bacteria and we will put it in different plates. And then we will see, we will put different plasmids, right? In different plates. I have prepared like already this plate. I have this bacteria, hopefully we'll see today together. I put it in the incubator, and then we will pick these bacteria and we will do the transformation with the plasmid. We will mix the bacteria with the plasmid, okay? And then we will incubate overnight, and tomorrow we will see if our hypothesis is true. And we will discuss if it happened or not. Okay, are you all clear and ready? Maybe we can like, I will go get the bacteria. I will connect from my phone. My friend, I think she didn't come. She had to help me with the camera.
Oh no.
She's not here, so and then maybe I can prepare the reagents, and when we come back from break, we can like do the transformation.
Yeah.
Let me just join from my phone. Where is this slide?
Yeah, if you guys have any questions.